Hi everyone, I'm Ziye from Sudan University. Today I will present our work on contact binning, MetaBinner, a high-performance and standalone ensemble binning method. The figure on the right shows the common metagenomic data analysis process. The yellow box marks three important metagenomic computational methods, namely assembly, contact binning, and taxonomic annotation. Here we focus on contact binning, which aims to group genomic fragments from the same genome into the same bin. Contact binning can help to explore new genomes and new genomic features. The commonly used binning methods combine chimeral frequency and abundance information, often better than those that use one kind of information. The most representative individual binning methods are ConCount, MaxBin, MetaBed, and the newly developed VMB. These methods take two kinds of information as input, obtain two kinds of distance or similarities through statistical models, perform clustering to get binning results, or concatenate two features for clustering. However, it is difficult for an individual binning tool to achieve ideal performance on different types of datasets. Therefore, we need to develop an ensemble binning method. There are two essential paths to an ensemble binning method. One is to get the component binning results. We hope these component binning results are diverse and accurate. The other part is the strategy to integrate these component results. We expect the integration strategy is efficient and effective. The ensemble binning methods can be divided into two categories. The standalone binners that integrate multiple different component binning results within the ensemble binner, such as BMC3C, and the binners that integrate the binning results of other contact binners, such as DS2, Binning Refiner, MetaRep, and UnitM. These methods usually use single-copy gene information to select high-quality bins from the output of different individual binners, utilizing the characteristic of the SCG, which are identified as a single copy in a large portion of the genomes within a specific phylum. The figure shows the workflow of MetaBinner. MetaBinner contains two main modules. Component module includes the first two false steps, used to generate high-quality and diverse component binning results. Integration module includes the fifth step, used to integrate the component binning results. We will present the details later. We get three feature matrices for each dataset. For some datasets with a large number of high-quality sequencing samples, good binning results can be obtained using only the coverage feature. In addition, for the datasets with high-quality context, good binning may be achieved with chimeral features alone. We kept feature matrices with coverage information only and chimeral frequency only. And in addition, we also get the X-Campbell metric that concatenates the two kinds of features. Similar to Coca-Cola, we utilized the set of single-copy genes universal for bacteria and archaea to estimate the number of genomes. So we determined the initial bin number K0 based on the number of contexts containing each SCG. A list of numbers larger than K0 was then sequentially tried as the bin numbers in the k-means. The bin number yielding the largest silhouette coefficient value of the binning results is chosen as the final bin number. To generate high-quality diverse component binning results, we proposed the partial seed strategy based on k-means plus, a variant of k-means. Let K denotes the bin number estimated by step 2. Instead of randomly choosing the K cluster centers, we use the SCG information to define cluster centers. For a particular SCG, we suppose that there are L contexts containing this gene. These L counties should belong to different genomes, which were used to initialize L cluster centers. Then we used the k-means ++ to generate the other k-L initial clustering centers and get the binning results. Since single copy gene set we used has multiple different genes, each ICG can correspond to a fixed set of initial cluster centers. To obtain different underlying clustering results, we selected 3 genes based on the number of contigs containing each ICG, obtaining 3 sets of initial cluster centers for each feature metric. In addition, we also run the regular k-means on each feature metric. In this way, MetaBinner generates 4 binning results for each feature metric, 3 from the parasitic method and 1 from regular k-means. Since each dataset has 3 feature matrices, we generate a total of 12 base binning results for the next step. To generate more bins with low contamination for further integration, we did post-processing for each binning result produced in step 3. We ran checkm for 1 binning result and obtained the contigs having the single copy genes. Similar to unit M, we then use the information to estimate each bin's contamination and completeness of each component binning result using the scaling strategy in checkm. Then, if a bin has high contamination and completeness, we split it by estimating the number of sub-bins of the bin using the same approach as in step 2 and run the k-means. To efficiently integrate component binning results, and considering that the quality of binning results obtained from different feature matches may vary significantly, we designed a 2-stage integration module based on Unit M and MetaWeb. In the first stage, we separately integrate the 4 component binning results generated in step 4 for each feature metric. First, the bins with high bin scores estimated by the ICGs will be selected. Then the contigs in the selected bins will be removed from other bins. The above 2 operations are repeated until there are no high-quality bins. In the second stage, we integrated the 3 ensemble results of the first stage, as shown in the figure. First, the binning refiner is applied directly to refine bins generated from the first stage to produce bins with low contamination level as done in MetaWeb. Then the same method as the first stage is used for selecting high-quality bins. We use 3 large-scale multi-sample simulated datasets from CMI and a real dataset for evaluation. For this real dataset, we used 2 advanced metagenomic assembly methods to obtain contigs. For the simulated dataset, we used EMBER for evaluation, and for the real dataset, we used checkm for evaluation. The right table shows the results of different methods on the simulated datasets. Under different completeness and contamination conditions, MetaBinner can recover more high-quality genomes than other binning methods, including individual binning methods and ensemble methods. Specifically, in terms of the number of near-complete bins, MetaBinner increased by 75.9% and 32.5% on average for the simulated datasets compared to the best individual binner and the second best ensemble binner, respectively. Take the CMI-GT as an example for analysis. Among the binners with the highest ARI, MetaBinner assigns the most base pairs among all the binners. MetaBinner achieves the highest average completeness of all predict bins. Its average purity is close to those of the binners with the highest average purity. This table shows the performance of different methods on the real dataset. MetaBinner and MetaRep achieve the best overall performance. Among the individual binners, ISMB achieves the best performance. As shown in the table, MetaBinner can recall the most near complete genomes. DS2 can also be used as an integration strategy. MetaBin A DS2, MetaBin B DS2, and MetaBin C DS2 denote the DS2 integration results of the component binning results generated using XCampbell, XCal, and XCOM respectively. This table shows the performance comparison in the CMI airways dataset. The proposed ensemble strategy has better performance than DS2 on all three feature combinations. For the datasets with more samples, for example, CMI Muscat, it takes much time to run MaxBin compared with other binners, so the whole running time of MetaBinner is much less than other ensemble binners. For the CMI airways dataset, the running time of MetaBinner is close to that of DS2. The earlier version of MetaBinner achieved the best overall performance on two of the three benchmark datasets used in CMI2 challenges. As for the third dataset, the MetaBinner recalls most high-quality genomes from its gold standard assemblies and hybrid assemblies. For an example binning method, component results should be accurate and diverse, and the ensemble strategy should be efficient and effective. To generate accurate and diverse component binning results, we propose the Petrol-seed method and use SCG and three types of features for binning. Further, we used an efficient two-stage ensemble strategy to integrate the component binning results effectively. MetaBinner achieves good performance on the CMI2 challenge and is freely available.
Okay, that's all. Thank you.
I'll start one. So yeah, that's very much good. So one thing concern is that we are using SGP criteria to do the binning, and then we use the same criteria to evaluate the binning performance. So these the question that the result might be overinflated, because that's like in machine learning world that's called overfitting, because the label information somehow got leaked. So I'm not well familiar with Amber. Does Amber also rely on SCG to measure the contamination and completeness?
I think that CheckM applies SCG to estimate the completeness and contamination, but Amber based on the gold assembly.
Okay. Yeah. So this also for some like unknown genomes that SCG may not work very well. So this might also limit the performance of MetaBinner. Is that right?
Yeah. I think that our binner perform pretty well on the simulated data sets, but the results on the real data set is good, but not that good. I think that which may be because that the quality of context influence the SCG estimation and for the Petrol-seed method.